This lesson is about enzymes, and enzymes are proteins that catalyze chemical reactions. In this image, the gray represents an enzyme, and the green represents a molecule that binds to the enzyme. And by the end of the chemical reaction, the green molecule has been broken down into two smaller molecules. Enzymes are a type of protein, and enzyme names usually end in ase, the suffix ase. For instance, amylase, lipase, and cellulase are all examples of enzymes. We should talk about a few vocabulary terms it would be helpful to understand. Optimal means best. For example, sunny skies and high temperatures are optimal conditions for a day at the beach. Those are the best conditions for a day at the beach. Or enzymes only function under optimal conditions. So enzymes have very specific conditions under which they'll operate, and we could call those optimal conditions, the best conditions for that enzyme. Rate means speed. For example, enzymes increase the rate of chemical reactions. They increase the speed of a chemical reaction. To catalyze something is to get it started. For example, enzymes catalyze chemical reactions. That just means enzymes get chemical reactions started. And a related word is catalyst. A catalyst is anything that catalyzes. So enzymes are catalysts. pH refers to how acidic something is. And the lower the pH, the more acidic. That's a little counterintuitive, but the lower the pH, the more acidic something is. For example, some enzymes require a high pH and others require a low pH. We might say that the optimal conditions for some enzymes are a high pH and the optimal conditions for other enzymes is a low pH. Enzymes increase the rate of chemical reactions. In other words, enzymes speed up chemical reactions. And the way that enzymes do this is they catalyze chemical reactions. In other words, enzymes get chemical reactions started. And because they do this, enzymes are catalysts. Think of a boulder at the top of a hill. It would take a lot of energy to push that boulder over the edge of the hill. But once you'd invested that energy, the boulder would then roll down the hill easily. And in that scenario, in that hypothetical situation, you'd be acting as the catalyst. You'd be acting a bit like the enzyme if you were the one to push that boulder over the edge of the hill and get the motion started. A chemical reaction is when a molecule is turned into a different molecule. The molecules can be combined or molecules can be broken down. And so another way of describing enzymes is to say that enzymes help combine molecules and enzymes help break down molecules. And breaking down molecules comes up more often. So if a question asks you about a substance that helps break down a molecule, it's probably asking you about enzymes. A couple more vocabulary terms. Specific. I'm not going to define it, I'm just going to give you an example and you'll understand it. My favorite part of the book occurred in the third sentence of the second paragraph on page 126. That's very specific. A non-example, an example of a statement that's not specific, would be, my favorite part of the book was the middle. That's a very general statement. That's not specific. Another word you want to understand is range. Range refers to the lower and upper limit of something. For example, it's between 83 and 85 degrees outside. That's a temperature range. Well, enzymes are very specific. Each enzyme requires a specific pH range. Each enzyme requires a specific temperature range. And enzymes only catalyze specific reactions. For example, maltase is an enzyme that catalyzes the breakdown of maltose, a carbohydrate. And pepsin is an enzyme that catalyzes the breakdown of proteins. But you can't swap these enzymes out for each other and get the same reactions. Maltase does not catalyze the breakdown of proteins, only maltose. And pepsin does not catalyze the breakdown of maltose, only proteins. That's what we mean when we say enzymes are very specific, or that's one of the several ways in which they're specific. How do enzymes work? How do enzymes catalyze such specific chemical reactions? The answer has to do with their shapes. Enzymes have very specific shapes. Remember, enzymes are proteins, and we said proteins have very complex shapes. And so the result is that enzymes have very specific shapes. Those shapes are designed for specific molecules. Not just any molecule can combine with any enzyme. And those molecules bind to the enzyme and react with each other. Or, as in the image below, a single green molecule binds with the enzyme represented in gray, and by the end of the chemical reaction, that molecule, represented in green, is broken down into two separate molecules. The point here is the shape of that gray enzyme. Notice that it matches perfectly the shape of the green molecule. What are some examples of enzymes? Well, one category of enzymes is digestive enzymes. And on the right, we have an image of your digestive system. The digestive system takes food and breaks it into molecules that can be used by your body. In your mouth, you have an enzyme called amylase. Amylase breaks down carbohydrates. In your stomach, you have an enzyme called pepsin, and pepsin breaks down proteins. And in your small intestine, you have an enzyme called lipase, and lipase breaks down lipids. These are just some of the enzymes involved in digestion, but it provides you with some examples of enzymes and what they do in your body. Another example of a digestive enzyme is cellulase. Now, cellulase is not an enzyme that humans produce. It's actually not even an enzyme that termites produce, although they require it. So a termite, as in this image, eats wood, and it digests wood. And it uses an enzyme called cellulase. This enzyme is actually produced by microbes that live in the gut of the termite. So there are tiny microbes that live inside this termite. And these microbes produce an enzyme called cellulase. And when the termite eats the wood, the enzymes produced by the microbes are used to break down that wood. Two other enzymes that we'll be hearing plenty more about are DNA polymerase and RNA polymerase. DNA polymerase is an enzyme used during DNA replication, and DNA polymerase is represented here in this image as these brown circles. But the DNA polymerase is being used to replicate or copy the DNA, so that's a very important protein. RNA polymerase is used to produce mRNA from DNA. You don't have to memorize these details right now, but just now you've heard of DNA polymerase and RNA polymerase, and these are two very important enzymes. Let's do some vocab and then we'll do some MCAS questions. Primary means most important. To hypothesize is to make an educated guess. Scientists hypothesize when they conduct experiments. And to secrete, it means to squirt out, to discharge, to emit. It's when something comes out of something else. For example, glands in our mouths secrete saliva. Or another example is cells in the stomach secrete pepsin. This one's a bit wordy. I'm going to explain it and just point out the answer. Scientists compare a specific type of enzyme from two species. One species is a bacterium that lives in hot springs. That's our first clue. This bacterium lives in hot springs. The other species is a shrimp that lives in cold ocean water. The scientists observed the activity of the enzyme from each of the species over a range of temperatures from 0 degrees Celsius to 100 degrees Celsius. Remember that enzymes require specific temperature ranges to function. Well, the enzymes in the bacterium are obviously going to require a higher temperature range than the enzymes in the shrimp. And so the answer to this question, I'm not going to read them all out, is going to be B. The bacterial enzyme has a different optimal or best temperature than the shrimp enzyme. The enzymes in the bacterium are going to require a higher temperature range than the enzymes in the shrimp because they live in very different environments, and enzymes only function under specific conditions. They require specific temperature ranges, among other things. Cellular respiration involves a series of chemical reactions. Which of the following is a primary way that enzymes affect these reactions? A, they decrease the pH of the products. B, they increase the rate of the reactions. C, they take the place of oxygen as a reactant. Or D, they change the location of the reactions in the cell. Well, the answer is B. They increase the rate of the reactions. We're talking about enzymes, and enzymes increase the rate of chemical reactions. Which of the following will most likely change when an enzyme is added to a reaction? A, the pH of the reaction. B, the rate of the reaction. C, the products of the reaction. Or D, the temperature of the reaction. Well, the answer again is B. We're talking about enzymes, and enzymes increase the rate of chemical reactions. Sucrase speeds up the breakdown of sucrose into glucose and fructose. Sucrase is an example of which of the following types of molecules? A, a disaccharide. B, an enzyme. C, a hormone. Or D, a nucleic acid. Well, the answer, of course, is B, an enzyme. We could infer this based on a number of things, but of course we have the suffix ase that helps give it away. And we are talking about the breakdown of a molecule, and that's also a hint. Enzymes are used to help combine molecules or to help break down molecules. Secretions from the pancreas contain compounds called lipases. Lipases increase the rate of digestion of lipids. Lipases are an example of which of the following? A, enzymes. B, hormones. C, nucleic acids. Or D, simple sugars. Lipases are an example of an enzyme because they increase the rate of a chemical reaction. And they also end with ASE. That's also a hint. Enzymes are a type of protein. And they're a type of protein that increases the rate of chemical reactions. It speeds them up. And they do this by catalyzing reactions. They are catalysts. And what that means is they get chemical reactions started. Enzymes require specific pH ranges and specific temperature ranges in order to function. And some examples of enzymes are amylase, pepsin, and lipase. These are involved in digestion. Cellulase is another digestive enzyme that exists in termites. They use it to break down plant material, wood. And DNA polymerase and RNA polymerase are two other examples of very important enzymes that we'll discuss in much more detail later. And notice that enzymes often end with the suffix ase. If you see ase at the end of a word, there's a good chance it's an enzyme.